Okay, here's my presentation on DNA replication. Some key terms to know are, well, first of all, you need to know what a nucleotide is, and that is a subunit of which the nucleic acids are composed. They're made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. The 4 types of bases are adenine, thymine, guanine, and cytosine. And adenine can only pair with thymine, and cytosine can only pair with guanine. Each of the bases are held together by hydrogen bonds. Adenine and thymine are held together by a double bond, and cytosine and guanine are held together by a triple bond. So you can see that right here in the rings of the ladder. The A is always paired with the T, and the C is paired with the G. And it doesn't show it, but there would be 2 in between the A and the T, and 3 between the C and the G. And then another key term to know is a double helix, and that's just the name for the shape of the DNA, which looks like a twisted ladder, like you can see in that picture there. And then the 5 and the 3 ends. So if you were to untwist the ladder of a DNA, it would look like a ladder. And the ladder has 2 parallel thick sides where you put your hands, and the steps would be like the DNA rungs of the ladder. And then each of those sides either has to be a 5 or a 3. And the 5 and the 3 always have to be opposite of each other. So if you were to use the ladder example, the top left could be a 5, the top right would have to be a 3, and then the bottom is opposite, so the left would have to be a 3 and the right would have to be a 5. So like here you could have this one be a 5 and then a 3, and then down here would have to be a 3 and then a 3 and then a 5. So it's always opposite. And then the top rail has to run 5 to 3 and the bottom has to run 3 to 5. The purpose of DNA replication is to ensure that each cell contains a copy of instructions and is able to function correctly within an organism. It occurs during the S phase of the cell cycle, so it goes G1, S, G2, and then it's either mitosis or meiosis, so the M phase. And this occurs during the S phase. Some important enzymes that are involved is the DNA helicase, and that is the enzyme that unzips the DNA, so it breaks down the hydrogen bonds so that they're each alone. So A is separated from T and G is separated from C. The DNA polymerase is the enzyme that joins the single unzipped nucleotides with their matching pair and forms a new strand of DNA. And then it checks the DNA for errors. So this is what brings the A and pairs it with the single A and pairs it with the T, and then the C with the G, or G with the C, or T with an A. And it checks the DNA for errors to make sure that it makes a normal kid, essentially. The DNA ligase closes nicks in the phosphodiester backbone of DNA. They are essential for the joining of the Okazaki fragments during replication and for completing a short patch DNA synthesis during the DNA repair process. And then the single-stranded binding proteins are proteins that attach to the single strand of DNA that's been unzipped from the double helix and keeps the strands from reconnecting to the double helix. So it just makes sure that when they are unzipped for replication, that they don't join back together, so that they can form with the other pair to make a copy of itself. And then the replication forks are the place on the DNA molecule where the unwinding of the helices and the synthesis of daughter molecules are both occurring, basically in the middle of the X or where the twisted ladder crosses. So here would be an example of where your replication forks would be, right in the middle of where they cross. Okay, so what happens in the replication process is that the DNA gets unzipped, like I said. So here, the helicase unzips it right here. So this is where it's at now because this has already been unzipped, and it will keep unzipping it so that each of the bases are separate from each other. So they all separate, and then the polymerase comes along and it repairs them up. So here you can see that these are adding right here, going this way, and then going this way. They're adding the other pairs so that it makes it a complete set of DNA. Yeah, that's basically how that works. And then so this will be an exact copy of this, so it'll basically be opposite. So you'll see here that they're getting paired with their opposite, the A's with the T's, the C's with the G's. Okay, so then the difference between the template strand and the new strand is that the template or the original strand may read something like the AC. Anyways, you can read that out, but then the new strand has to be the opposite, so it would be TA, so the A would switch to a T because it's opposite and it has to pair up. And if the original strand is 5 to 3, then the new strand has to be 3 to 5. The direction of the replication always has to go 3 to 5, so that when it reads 5 to 3, like in the bottom strand, it has to jump forwards, work backwards. So like down here, or up here actually, it's having to go this way and then work backwards. Come this way more, work backwards. Come this way and work backwards, so that it can read in the right way, because it's not the 3 to 5 like it's supposed to be. And then the leading strand of DNA is the strand of DNA that's being synthesized in the same direction as the growing replication fork. The lagging strand of DNA is the strand of DNA whose direction of synthesis is opposite to the direction of the growing replication fork. So it is synthesized in short separated fragments, which basically is what I said. It jumps and then works backwards, jumps and works backwards. And then the Okazaki fragments are the newly synthesized DNA fragments that are formed on the lagging template strand during the DNA replication. And then the end result of the replication are two identical double helixes of DNA, so it's an exact copy of the original DNA you unzipped and copied. And then those are my sources.